Hello, I'm going to talk about the epithelial barrier theory. It is our 25 years of work describing the development of allergic, autoimmune, and many chronic inflammatory diseases. Part 1. The pandemic of allergic, autoimmune, and neuropsychiatric diseases and multimorbidities. This is a time course of allergic diseases, starting from 1819 with the first demonstration of John Bostock and the first book of Charles Blakely on hay fever and hay asthma, in which he collected the first 28 cases, and it took him 3 years to collect these cases. And at that time, lawn making, house heating, increased grass pollen exposure were mentioned to play a role in the development of allergic diseases. But their prevalence was very low, and the diseases were mainly existing in big cities. And here we have a big change starting from 1960s, because we realized that hospitalizations increased in UK, and this was followed by Australia, UK, New Zealand, Canada, and USA. And these were patients coming from inner cities, and the hospitalizations increased mainly in the capital cities of these countries. And we realized that there was rising incidence in the soldiers, also coming from the cities, not from the countryside. We come to 1989. This is the reunification of Germany. And here, in the beginning, East Germany had the prevalence of asthma and atopic dermatitis around 1 to 1.5%. And West Germany had around 10%. Within 10 years, in the year 2000, East Germany levels reached to West Germany levels, with the children becoming sick and developing asthma, atopic dermatitis, and allergic rhinitis. Here we see the first curve, and this is increased prevalence of allergic diseases. But we realize later that autoimmune diseases are also increasing at the same time, and metabolic conditions. And there's the obesity pandemic, and there's type 2 diabetes, rheumatoid arthritis, multiple sclerosis. And this was happening in the industrialized countries. Then we have a second curve related to food allergy, peanut butter sensitization and anaphylaxis, and eosinophilic esophagitis. And this second curve starts around 2000s. And here we see WHO numbers. In 1960s, there was 30 million patients with chronic non-communicable diseases, which increased to around 2 billion patients with 100 times increase. At the same time, world population increased by 2 times. That means there was a net 50 times increase in the prevalence of these diseases. Then we have another curve. This is the curve of big developing countries, mainly China. For example, asthma was 0.8% in 2000, and nowadays we are talking about 10% asthma prevalence in China. So the epithelial barrier theory includes the barrier damage, microbial dysbiosis, and immune system activation. And our work in this area started with the first studies with Axel Trautmann on skin and lung barrier in eczema and asthma. In around 2006, we published in Current Opinion Immunology first highlights of the epithelial barriers and understanding of the epithelial barriers to keep away the factors, to wash the factors if there's too much inflammation, and to suppress around the epithelial barriers. Then we identified immunological effects that open or close the epithelial barriers. And then we started to focus on the environmental change, exposome change, and toxic substances. So when we are talking about gut-skin axis, skin-lung axis, gut-brain axis, gut-lung axis, lung-brain axis, they are all happening in the epithelial barrier theory. And one organ is inflamed, and the inflammation is carried to other organs and makes them sick. And the old theories like hygiene hypothesis, danger hypothesis, old friends, ignorance, these hypotheses are all in the epithelial barrier theory. They are covered, and epithelial barrier theory is an umbrella theory that covers all the possibilities related to immune system activation, microbial dysbiosis, and epithelial barrier damage. We tried to identify the diseases related to the criteria, and our first criteria was increased prevalence of the diseases after 1960s or after 2000s, which is not affected with the changing method of diagnosis. As an example, when you look at eosinophilic esophagitis, when we were in internal medicine, we were making endoscopies, and then hematoxin and eosin staining was sufficient. Or when we were doing ultrasound, when you touch the probe to the abdomen, you can easily see that there is fatty liver within a few seconds, and it's still the same method of diagnosis. So the diagnostic method stays the same here, but the prevalence shows a very significant increase. The second criteria is epithelial barrier defect and epithelitis with the activation of inflammasome, release of alarmins, interleukin 25, interleukin 33, TSLP, and multiple chemokines. And the third criteria is microbial dysbiosis is always associated with the immune system activation or epithelial barrier damage or epithelial cell activation. And this is characterized with the loss of commensal bacteria and colonization of opportunistic pathogens. For example, Staph aureus colonization in atopic dermatitis, in chronic rhinosinusitis, Borexella, Haemophilus pneumococcus colonization in asthma. And then the fourth criteria is the pandemic of multimorbidities, different and practically irrelevant diseases that fulfill these criteria appear in multimorbidities. That means in our old times when we were assistants, we were seeing these diseases as single entities, but currently we are seeing them together. So an asthma patient develops colitis and also has chronic depression at the same time, or an allergic rhinitis patient develops atopic dermatitis, but at the same time has a chronic eye disease or chronic heart disease. And the fifth criteria is circulating inflammation. That means we are seeing visibly by our new methods, by doing deep omics analysis of the circulation, looking at the cells there and looking at the inflammation that carries the inflammation from one organ to other. So with this way, we identified more than 70 diseases. And this is a further reading. It's a very detailed article with the involvement of worldwide experts of many organ disciplines. And here we can see that we have the diseases related to skin, which fulfill these criteria: atopic dermatitis, alopecia areata, allergic contact dermatitis, irritant contact dermatitis, psoriasis. And in the airway group, we have asthma, allergic rhinitis, chronic rhinosinusitis, COPD, and many others. In the digestive tract diseases, we have eosinophilic esophagitis, periodontitis, food allergy, inflammatory bowel, celiac, and many other diseases. The neuropsychiatric group has Alzheimer's, Parkinson's, autism spectrum disorders, stress-related psychiatric disorders, chronic depression, ischemic stroke, migraine, multiple sclerosis, amyotrophic lateral sclerosis. The autoinflammatory and metabolic conditions is a big group with rheumatoid arthritis, osteoarthritis, diabetes, obesity, metabolic dysfunction associated with cirrhotic liver, that means fatty liver. And we have here autoimmune hepatitis, thyroid diseases, systemic lupus. And we have ocular diseases with increased prevalence, age-related macular degeneration and glaucoma, dry eye, uveitis, and also allergic conjunctivitis, and other diseases, and end-stage renal diseases, heart failure, myocarditis, osteoporosis, anemia, hemolytic uremic syndrome, sepsis, severe COVID, cardiovascular diseases, polycystic ovary. They are all involved with these criteria. Another important paper which I'll mention here is the One Health concept, because pets and domestic animals are also showing increased diseases, and that's an important thing because of their very high exposure to mainly pet food, which is packaged food containing many emulsifiers and additives, and also they are exposed to air pollution like humans, or they are exposed to microplastic like humans. So this has made a big financial burden, of course, increasing the chronic non-communicable diseases to around 2 billion patients. And it affects the GDP of many countries, and health expenses in the world average increased to around 10%. And in the US, in 1990, only one single disease, asthma, was costing $10 billion. Currently, it is around $150 billion. And in the next years, $2 trillion is needed for asthma in the United States and Europe. So one single disease will cost a huge amount of money. Yes, the non-communicable diseases cost more than $10 trillion per year in the world, and about 17.6% of the GDP is used for health in the US in 2024. Part 2, exposure to epithelial barrier damaging substances. What we are seeing here is the epithelial cells of a patient, or even 1/3 of healthy individuals, that the epithelial cells of the skin and mucosas, lung, bronchus, sinus, nose, and gastrointestinal mucosas are not touching each other. And they are affected by the environmental toxic substances, and the microbes pass through and even to deeper areas. So when we list these substances, we have laundry and dishwasher detergents and rinse aids, shampoos and body cleaners, household cleaners, surfactants, sodium dodecyl sulfate, sodium dodecyl benzene sulfate, and toothpaste containing these surfactants. The second group is enzymes and emulsifiers in processed food, food colorants, sweeteners, and high glucose. The third group is air pollution group with cigarette smoke, ozone, diesel exhaust, nanoparticles, micro and nanoplastics, and particulate matter. And then these barriers are open. Microbiome goes deeper in the tissues. This is called bacterial translocation, and the diseases are starting. And these factors have a synergistic effect between each other. Just think of a patient who is house dust mite allergic, but there is a cleaning day at home, and the cleaning material has affected the respiratory system. And at the home, the packaged food is consumed, which has enzymes and emulsifiers in it. And then there's someone smoking cigarette at home. So think that this is a very bad condition that may cause asthma exacerbation. Or there is particulate matter that they are living next to a main street, or diesel exhaust exposure. So these are all factors that work together to cause the inflammation. The dose and cumulative effects are essential here for these substances. I would like to quote here Paracelsus, the pharmacist of Canton Schwyz in Switzerland, and Paracelsus in 1520s has said, "Sola dosis facit venenum." So the dose makes the poison. And all things are poison, and nothing can be without poison. But only the dose makes that a thing is poison or the thing is a no poison. So we continue here. Part 3, introduction of sodium lauryl sulfate, sodium dodecyl sulfate, and enzymes to detergents in 1960. We see it as STS and SLS. And 1960 is a milestone time because these additives were added to detergents, and the detergent industry made a big change by making all the clothes very clean, and the laundry was quite successful for the cleaning effect. And sodium lauryl sulfate in the beginning was added around 20%. Now it's decreased to 10%. And it is in many detergents, there is 10% sodium lauryl sulfate. In shampoos and household cleaners, you can see 5 to 10% sodium lauryl sulfate. And in some toothpaste, after 2000, we are seeing that sodium lauryl sulfate is added in the range of 3%. And it can be extracted from house dust, pillows, bed sheets. It is toxic to cells, and we can see it in organoids, organoid chips. And the dose of toxicity is 1 to 50,000 times. And there's in vivo studies, and the toxic threshold is 0.002%, whereas it is included in these detergents and toothpaste around 3 to 10%. So for this area, please read the papers of Hideaki Morita, Arturo Rinaldi, Mu Xian, Ming Wang, Yasutoka Mitamuro, Alfred Doyle, and Benjamin Wright. So here we have the 50,000 times diluted sodium lauryl sulfate or laundry detergent that affects the epithelial barrier. What we are seeing in the transcriptome is that there is upregulated lipid metabolism, oxidative stress, endoplasmic reticulum stress, cell survival signals, and interleukin-33 and alarmin. What is downregulated is cell adhesion, extracellular matrix organization, wound healing, and DNA repair. So the interpretation of this finding is a chronic wound that tries to survive the cells, and it's a carcinogenic environment because of endoplasmic reticulum stress and DNA repair defects. So here is a very exciting study coming from Hideaki Morita and his group. And here we can see that only 4 drops of a detergent, or in the same study we can see sodium lauryl sulfate also, added to the nose of mouse, goes to respiratory system and cause an eosinophilic bronchitis. And peribronchial, very strong eosinophilic asthma-like model. And we can see total leukocytes increase in the lungs. Eosinophils shows a very significant increase, and there is some neutrophils in addition. And here we see IL-5 reporter mouse, which shows that mainly type 2 innate lymphoid cells are the main source of type 2 cytokines, making interleukin-5 in around 44.4% after detergent exposure, and this is the control. And basophils and T cells have no effect. Then we looked at what stimulates the type 2 innate lymphoid cells, and here we can see that detergent-exposed lungs have a lot of interleukin-33 production. And here we have interleukin-33 knockout mouse and wild-type mouse, and we can see that the total leukocytes do not change, but eosinophilia is increasing with the wild-type mouse, which decreases to normal levels in interleukin-33 knockout mouse. So the story is interleukin-33 coming from the epithelium after detergent exposure activates the type 2 innate lymphoid cells, and this makes eosinophilia. And this is an interesting study also coming from the group of Hideaki Morita. And here we see that O-albumin-specific IgE is induced in detergent-exposed mouse. And this is a very interesting finding. And if we have the antigen alone, there is very little IgE. But if there is a detergent exposure at the same time, then we can see that IgE is going very high. And this is one of the first findings showing the induction of IgE with an environmental exposure, which can be the reason for the development of allergic diseases. We are using ex vivo human skin models coming from the group of Nicolas Gaudenzia, and we have made several papers. And we are also using an electric impedance spectroscopy, which is developed by our group. And this is mainly used to detect skin barrier. And with one touch, within 2 seconds, you can detect the skin barrier. And we have published several nice papers on this. And we have used here in this model, by using these 2 devices and by exposing to 2 bacteria, 2 very healthy probiotics, Bifidobacterium breve and Lactobacillus reuteri. And we can see that by extreme gradient boosting, XGBoost regression model, it's a machine learning model, we identified most critical genes associated with Bifidobacterium breve induced barrier enhancement. They include OL2, keratin, and ETV4, LAT-S2, Notch, and MH515, and cyclin-dependent kinase, and Claudin-10, a barrier molecule. So we are now in the way to identify what makes strong epithelial barriers and how can we modify these genes. And I would like to mention this paper. It's about development of eosinophilic esophagitis. Eosinophilic esophagitis was first described by Hans-Uwe Simon and Alex Straumann in our institute. Actually, the paper is coming from SIAF. And in this paper, they show the first series. There was single cases mentioned before this. And in this study, Alfred Doyle and Benjamin Wright showed that STS, sodium dodecyl sulfate, which is the main culprit in the detergent and cleaners and also several cosmetics, is stimulating IL-33 production from esophageal epithelial cells, and we see very high increased IL-33. And this is STS-exposed esophageal epithelium and esophageal tissue. You can see a strong cellular infiltration here and thickening of the mucosa, and there's a lot of eosinophils increasing and infiltration of eosinophils. And this is for comparison, a normal esophagus. And we can see that in this paper, exposure to STS causes decreased epithelial barrier integrity, epithelitis with increased IL-33 production, eosinophilia, and inflammation, abscess formation, and CD4 positive T cell infiltration and remodeling in the esophagus. And this is STS and SLS, sodium dodeoxysulfate, is the main culprit that comes from the toothpaste and household cleaners to make eosophageal esophagitis. So what are our suggestions? Now, a group of doctors are working to find evidence and to suggest to patients to avoid these substances that are causing allergic diseases and autoimmune diseases. And we have SLS, STS and enzymes of detergents, cosmetics, and cleaning substances. And here we are suggesting to use an extra rinse and a small amount of detergent and opt for liquid detergents instead of powder. Choose SLS-free products. Read ingredient labels. Use natural or organic products to limit the use of foaming products, because the foaming products are changing the surface tension and then they are opening the epithelial barriers and initiating inflammation. Avoid prolonged contact. Usually less than 1 minute contact can be tolerated, but when the contact is long, we have data with 1 minute and also 5 minutes, the inflammation becomes very significant. And be cautious with heat, because heat also opens the epithelial barrier. Hot water opens the epithelial barriers. Together with these toxic substances containing cosmetics and detergents, the damage increases. You can look at the website epithelialbarriertheory.com, and there's a very nice app, Yuka, and this app describes almost all the substances. There's more than 2.5 million substances mentioned in the Yuka app. And you can identify whether the cosmetic or the substance or the food you are using is damaging the tissues, and together with the literature. Now we go to gastrointestinal system. Part 4. Professional dishwasher rinse aid, alcohol ethoxylate. These very shiny glasses were existing in our household. And in around the year 2000, there was a big revolution in the dishwashing, and professional dishwashers became very common in the restaurants, hospitals, dormitories, and armies. And these very shiny glasses became part of our life. And these shiny glasses are obtained by washing with these professional dishwashers, which has a washing time of only 1 to 2 minutes with 82 degrees and 4 atmosphere pressure. And there is no rinsing. In the last 20 seconds, the toxic rinse aid is added and it is dried and it is shining on the wash material and it can be extracted. So we performed experiments to extract this toxic culprit, alcohol ethoxylates, and we identified that it is toxic in the 1 to 20,000 dilution. And we showed that the dishwashing residues, after dishwashing, we extracted it from dishes, cutlery, glasses, and we can see that it has showing cytotoxicity still when we extract it with medium, and further dilutions to 1 to 5, 1 to 10 is showing cytotoxicity. And we can see that it is damaging the epithelial barriers with 1 to 2, 1 to 5 dilutions, and it is causing the passage of FITC dextran with these doses. Please go through this paper by Oler et al. In Journal of Allergic Clinical Immunology. So suggestions to avoid alcohol ethoxylate, because alcohol ethoxylate is also nowadays in household detergents. The bullet in the middle has alcohol ethoxylate, and there's this toxicity in 1 to 20,000 cannot be rinsed away with normal rinse. So it will be good to choose alcohol ethoxylate-free products. Do not frequently change plates and glasses when you are going to a restaurant. And because every time you are having another one with another shining plate, shining glasses, and you have to rinse them with water before use. Limit the use of rinse aids at home or also in the public areas, and read labels carefully, and it's possible to use eco-friendly or plant-based dishwashing. Then we have here part 5. Processed and packaged food, emulsifiers, colorants, sweeteners, and antibiotic-like substances. So packaged food was introduced to our lives in around 1980s. But after 2000, it became very common. When we were going to supermarkets around the year 2000, they were changing the shelves every 7 days, because the pizzas, the sauces, had a shelf life of only 7 days at that time. But the same pizzas and the same sauces nowadays, when we go to the same supermarket, we are seeing that they have a shelf life of 1 year. So what happened after the year 2000? The commercial aim was to extend the shelf life and prevent the separation of the liquids. So to keep the liquids inside the pizza or inside the packaged food was very important, because if it was giving out the liquids, it was looking dirty. Nobody was buying it. So then there was food additives and emulsifiers added, and it was needed to kill the fungus and bacteria growth with very long shelf life. So there were substances that have antibiotic effect were added, and a thickening effect was observed, because with the thickening effect, the shelf life showed a very significant increase. So here is the list of emulsifiers used in USA and Europe, and we will mention we have worked with some of them, and we will mention now the effects of this datum, diacetyl tartaric acid esters of mono and diglycerides, lecithin, polysorbate 20, polysorbate 80. Polysorbates are twin that are commonly used in lab. They are so soapy. And here is polysorbate 20 and polysorbate 80, and epithelial barrier disruption caused by them. Polysorbate 20 and 80 are used in food, bread, ice cream, around 1% in ice cream, confectionery, beverages, and dressings. But we identified by using stem cell-derived intestinal organelles. Colon organate organelle chip. It's like a small colon, which is 3-4 millimeters, and liquid interface cultures. And we can see that the epithelial barrier is damaged, transepithelial resistance decreased, and paracellular flux increased. And here we can see the doses of polysorbate 20 and polysorbate 80 damaging the epithelial barriers, and the allowed dose is 1%. And we can see here doses that are lower than 1%, like 10 to 20 times lower doses are damaging. And we can see that 20 times lower concentrations are affecting the molecules here in the proteomics. We can see that interleukin 4, interleukin 6, interleukin 8, interleukin 13, interleukin 24 are stimulated by polysorbates. We see 4 chemokines here: CXCL1, CXCL5, CXCL10, CCL11, and one alarmin, TSLP, TGF-alpha, TNF, IL-22 receptor. They are all induced. That shows that these polysorbates are causing an inflammatory environment. Part 6. Why do we have peanut anaphylaxis? Humans were consuming peanut for so many years, and there was no such disease written in the textbooks before the year 2000, and after 2003, the anaphylaxis to peanut started. So we looked at what happened to peanut butter after 2000, and we identified that 2 emulsifiers. One of them is datum, diacetyl tartaric acid esters of mono and diglycerides is added. The other one is soy lecithin, and soy lecithin and datum were included in the peanut butter in the year around 2000, and they are the most common peanut butter emulsifiers. And we can see that they are decreasing the epithelial barriers, and this is gut on a chip. It's like a small organ, and colon organate on a chip, like a colon in the chip, and we can see that these doses are significantly decreasing the epithelial barrier integrity, and very strong decrease we can see with 1 milligram per ml, which is 1 to 1,000, 4 milligram per ml, and also 0.24 milligram per ml, which is 1 to 4,000 dilution, are decreasing the epithelial barrier, which is something that we didn't want. Then we made a mouse experiment, and we fed the mouse once a week for 6 weeks, and here we see the effects of gastrointestinal inflammation. Since there is a rapid passage through the esophagus, the inflammatory genes are a little bit low. In the stomach, they increase. In the ileum, they increase. And in the colon, they are much more. And this is with soy lecithin. And we can see here IL-33, TNF, CCL17, and aryl hydrocarbon receptor, and an oxidative stress molecule, PRDX5. They are all increasing in response to soy lecithin. And here we can see gastrointestinal inflammation by datum, and these are the substances like tumor necrosis factor alpha, CCL17, aryl hydrocarbon receptor, again PRDX5 are increasing, and esophagus, stomach, and the inflammation increases in ileum, and much more inflammation in the colon by datum. that is included in the peanut butter starting from the year 2000. And what is very interesting and exciting is that we have here two IgG and Ig induction data, and IgG is induced by soy lecithin, and we see here IgE is also induced by soy lecithin, total IgE. And this is a human study, and the data stimulates with an IL-4 induced IgE, again, 2 times more. And IL-4 induces IgE, which is very normal. It is the main cytokine to stimulate IgE. And here we can see a dose-dependent effect of datum in human PBMCs, increasing the IgE mRNA. So suggestions to avoid packaged and processed foods, additive and emulsifiers. Read labels carefully. Avoid most of the processed foods. Choose whole foods. Focus on fresh fruits, vegetables, whole grains, and unprocessed proteins to minimize exposure to emulsifiers found in packaged items. Cook at home. Avoid colorants and sweeteners. Opt for natural alternatives. Part 7. Micro and nanoplastics. It is very important to mention micro and nanoplastics here because they are produced in the range of 9.5 billion tons plastic production happened, and per year there's around 400 to 500 million ton produced, and there is 7 billion ton of this plastic remains as pollution. And we can assume that there is 1 billion ton microplastic that is in our soil and in the water and also sometimes in the air. And plastic has been found in amniotic fluid, pericardial effusion, coronary artery plaques, brain, testis, ovary, and plastic is ingested by the cells. It is immune modulatory, epithelial barrier opening, and receptor binding. And here are several studies coming from our group on plastic, and a summary of these studies in 4 points. Small nanoplastic, 0.1 to 1 microgram, is more dangerous as it goes inside the cells and affects many pathways. Method of plastic detection has changed. About 5 years ago, with the detection sensitivity of 1 microgram, we were saying that many PET bottles or many places are free from plastic. But currently, by using atomic force microscopy and infrared spectroscopy, 10 nanograms is detected, and this 10 nanograms is found in the brain, testis, ovary, epithelial fluid, meconium, so also in PET bottles. Plastic is detected in all organs. Its detection in organs such as brain, coronary arteries suggests that tissue barriers are not capable of protecting key organs against plastic. And a very important point is exposure to plastic pollution is together with bisphenols, BPA, BPS, phthalates, parent polyfluoroalkyl substances, PFAS, and dioxins. Probably you have heard that all of these substances are very cancerogens. So suggestions to avoid micro and nanoplastics are here. Drink filtered water, nanofilters, and avoid using PET bottles. Pay attention to the selection of meat and fish. Use products from large seas, clean seas, and high-altitude sources. Avoid skin and organs of seafood. Replace plastic tea bags with loose-leaf tea. Do not open big plastic packages inside your house because it goes to air, then you quickly inhale the plastic. Fully avoid plastic utilities in the kitchen. Use synthetic fabrics in your dresses. Wash with cold water. Consider microfiber laundry bags to capture released plastic. And the plastic area is very open for inventions. So just remember now these timelines with the increased prevalence of allergic autoimmune disease, with the second curve, and with the third curve. And here we are putting certain milestones. The first one is in 1960, addition of sodium lauryl sulfate, sodium dodecylbenzene sulfate, enzymes to household detergents and cleaners. The second timeline is starting from the year 2000 with increased food allergy, peanut butter anaphylaxis, eosinophilic esophagitis, additives and emulsifiers in processed food and toothpaste. And the third one is micro and nanoplastic, which will be probably a forever problem because of the very high pollution And it's a matter of the United Nations, and the whole world should be very cautious about plastic and its effects because of its immune modulatory and cell-destructive effects and changing the cell metabolism effects. These are all happening, and we are showing it, and many other groups are showing it as well. Part 8, molecular mechanisms of cellular toxicity. So what makes the toxicity when we are exposed to these substances was an important research area in our group, and I'll show you some data. This is about aspartame, and aspartame has a warning from WHO. It is being used for more than 30 years, and it is still around, and we can see that in human intestinal organoids derived from the stem cells, aspartame with daily exposure doses 2.5 milligram per ml, or 10 milligram per ml elderly with high doses can induce a toxic effect and inflammation. And this is a colon line induced showing NF-kappa B reporter, and NF-kappa B is induced. And oxidative stress with aspartame starts with relatively low doses, 2.5 milligram per ml, and we can see that it is increasing with increasing doses. And this N-acetylcysteine, an antioxidant molecule, is antagonizing the oxidative stress induced by aspartame. And here we see the cellular cytotoxicity to organoids. Aspartame-induced organoids become small and they die within 2 days. And here is the transcriptomic effect, cytoplasmic translation, response to unfolded protein, chemical carcinogenesis, positive regulation of apoptotic signaling, TNF signaling pathway, response to oxidative stress, leukocyte migration, DNA replication, double-strand DNA break. These are the downregulated molecules. So DNA replication is also hampered, and the DNA cannot repair itself, which is a very important molecular mechanism to avoid cancer development. And here we can see the DNA repair mechanisms defect, and these are the DNA repair-related genes, and aspartame is stimulating them with those. And the paper just appeared in Allergy last week. And here is a loss of mitochondrial membrane potential, which is very important. We have to protect our mitochondria against these toxic substances. And here is a JC1 staining, and this shows the effect of polysorbate 80, and the mitochondrial damage is happening with very low doses. You look at this dose. 1% is allowed from polysorbate 80, and this is 0.02% volume per volume, and 0.04%. And this is around 1 in 2,000 and 1 in 2,500 and 1 in 5,000 times dilutions. And these dilutions, which is around 50 times lower than the allowed dose, is showing the mitochondrial damage and change in mitochondrial membrane potential. Another important thing here is shown with polysorbate. You can see the dots of mitochondrial DNA breaks, which is controlled and which is polysorbate treated. As you can see, it is doubled or tripled the yellow dots here showing the mitochondrial DNA breaks. And here we can see the data, a very clear increase in the mitochondrial DNA breaks, and that's happening in the mitochondria. In the nucleus, it is not so significant, but there are some effects. But in the mitochondrial DNA breaks and mitochondrial toxicity with polysorbate is very visible. So in summary, mechanisms of epithelitis and epithelial barrier damage is here listed. What we have observed from our own research, which is done by Pat, Yazıcı, Babayev, Mitamuro, Zeynaloğlu, Bichel, Oğur, Lee, Yao, Yavrino, and we can see there is oxidative stress, which is coming together with the cytoplasmic panic in these cells, endoplasmic reticulum stress, and production of unfolded proteins, and mitochondrial stress, as I have shown you, mitochondrial DNA breaks and mitochondrial membrane potential changes, delay or inefficiency in DNA repairing, and cell death by apoptosis and ferroptosis. Together with this, there is microbial dysbiosis taking place and multiple effects of microbial dysbiosis. It's a huge topic on its own. We see inflammasome, alarmins, and multiple chemokines in response to these substances, and innate and adaptive immune activation. So overall, this is a full regulation of the immune system and effect on the cells, even leading to cell death, loss of function of the cells, a very severe stress, happening with some of the substances in daily use doses. Part 9, conclusions. This is an interesting slide, which is quite philosophic, because it's showing that the homeostatic interaction of epithelial cells and immune system with commensals is broken. We are carrying about 1 kilo of commensal bacteria in our body. And when the first eukaryotic cells developed around 1 billion years ago, they decided to carry some prokaryotic cells, and also the mammalians are carrying prokaryotic cells, and using the nutrients, vitamins, metabolites, using them for their digestions, immune education, cell differentiation, tissue maturation. And this interaction is now broken since 1960s, and Homo sapiens is responsible for this. And in the gastrointestinal system, there was around 4,000 species, which is now decreased to around 2,000 and even less in patients. And skin, it has 800 species, which is decreased to around 300 species of bacteria. So this is showing here a very clear relationship of epithelial barriers with biodiversity hypothesis, because when the epithelial barriers are leaky and when the immune system is activated, and then opportunistic pathogens are colonizing, and the pathogens are pushing away the healthy bacteria, and we are losing our healthy bacteria in our gastrointestinal system. So this is the first event taking place when we are exposed to these substances, and the epithelial barriers open, and microbiome pass in between the cells and to the deeper, translocate deeper. And this is together with microbial dysbiosis. Opportunistic pathogens colonize Staph aureus, Moraxella, Haemophilus, and Pneumococcus. And we have here IL-1, IL-25, IL-33, TSLP, and multiple chemokines. They initiate the inflammation, and the immune system comes to the area. So when the immune system comes to the area, the disease starts. It can be colitis, it can be chronic rhinosinusitis, it can be asthma, or it can be rheumatoid arthritis, that the gut is inflamed, but it's visible in the joints. The second event is the expulsion response. So the body is not happy that these microbes are too deep. That may cause sepsis. And to avoid sepsis, expulsion of microbes, allergens, toxins, pollutants is taking place. And the immune system is trying to push, because of the tissue pressures, which is around 5 to 15 centimeter water, the tissue pressure is pushing the microbes away. So this is an expulsion response, but at that time, we are seeing the diseases very clearly. And there is a detectable autoimmunity and neuroimmune interactions is taking place, and a chronic inflammation. So to summarize all of these findings, we have the direct toxicity to epithelium and microbiome, disrupting and opening of the barriers. And epithelitis with microbial dysbiosis takes place, and production of alarmins and multiple chemokines. The expulsion response is together with the efforts for the expulsion of the inflammation and deep translocated microbiome. And then we see circulated microinflammation and multimorbidities, migration of inflammatory cells to distant organs. And finally, because of epigenetics, chronicity takes place, and the epithelial barriers cannot heal. And also many cells are affected with these epigenetics. The dendritic cells around the epithelium, innate lymphoid cells, they're all affected, and the epithelial barriers cannot close, and the chronic disease may continue. And I finally would like to show some key papers. And there was a movie in Arte channel. Articles were published in many top newspapers, more than 400 newspapers, and more than 1,000 websites. You can reach the epithelial barrier, the theory.com website, and there's continuous information and update of the barrier, and continuous publications are coming from our group. I thank very much our international collaborators and the group members. And I would like to invite you to our World Immune Regulation meetings to discuss the epithelial barriers in a better way. Thank you very much for your attention.